Good afternoon, everyone. Welcome to the HPO seminar this week. Today, I am very excited to announce that we have a guest speaker. Eric, could you give us an introduction about our speaker, please?
Of course. Today, it is my pleasure to introduce our speaker, Dr. Greg Sawicki. Dr. Sawicki is an associate professor at Georgia Tech with appointments in the School of Mechanical Engineering and the School of Biological Sciences. He holds a Bachelor's of Science from Cornell University and a Master's of Science from the University of California, Davis, both in mechanical engineering. Dr. Sawicki completed his PhD in human neuromechanics at the University of Michigan, Ann Arbor, where he researched power ankle exoskeletons. He then went on to a National Institutes of Health funded postdoctoral fellowship in integrative biology at Brown University in order to understand how muscle physiology could play a role in the efficacy of exoskeletons. Dr. Sawicki now directs the Human Physiology of Wearable Robotics Laboratory, where the goal is to combine tools from engineering, physiology, and neuroscience to discover neuromechanical principles underpinning optimal locomotion performance and to develop lower limb robotic devices capable of improving both healthy and impaired human locomotion. By focusing on the human side of the human-machine interface, Dr. Sawicki and his group have begun to create a roadmap for the design of lower limb robotic exoskeletons that are truly symbiotic. That is, wearable devices that work seamlessly in concert with the underlying physiological systems to facilitate the emergence of augmented human locomotion performance. Outside of the lab, Dr. Sawicki enjoys spending as much time as possible exploring the outdoors by hiking, biking, skiing, and snowboarding when in the North or West with his partner, Katia Cole, an infectious disease biologist, and his daughters, Elodie and Sonia. Dr. Sawicki also seeks out music experiences whenever possible and never misses a Phish concert within the radius of a single day's drive. I will now give the floor to Dr. Sawicki for his talk on looking under the skin to understand human exoskeleton interactions during locomotion.
Thanks, Eric, for that great intro. And thank you guys for inviting me. I hope the connection's gonna remain stable. I am enjoying the outdoors with my girls this week. We're on spring break at the beach. And so I'm on a cell signal rather than a stable Wi-Fi signal. So if you lose me, please call and we'll try to get back. Let me go ahead and share my slides here. Let's see. Can you guys give a thumbs up if you can see those? And then I'm going to go into presentation mode. There we go. Let me close this. And I'm going to bring a pointer up here. Can you guys see the pointer? It's like a red blob.
Yep.
Okay.
Everything's good.
And can you guys still see me? Looks like maybe not. Now I'm back. Okay. Great. So this is just the title slide. Like Eric said, I'm going to tell you today about how we've been using non-invasive imaging tools to look under the skin when people walk with exoskeletons, to try to understand kind of the muscle level effects, the muscle level mechanisms for how these devices influence metabolic energy costs during movement. All right, let's see if we can advance. There we go. I like to usually start off with this kind of broad overview of how we think about research in the lab. Physiology of wearable robotics really is trying to learn from physiology to design better robotic devices, but also in turn to understand physiology a bit better by putting devices on people and studying the co-adaptation and the combined movements. And we like really simple models. even if we don't write the math out and simulate these. This is kind of the cartoon that typically we have in our head in the group. This looks like a long compliant muscle tendon unit because we've mostly focused on the ankle joint up to this point in the lab. And what you can see there is we've got a muscle tendon unit, a lever, some force that's being placed on the world to propel this thing, and a little bit of inertial dynamics and some gravity. And the way we think about assistive devices really are just forces in parallel with muscle tendon units. And you can think about that in the simplest way at a single joint like this. And what we've been trying to understand, not just us, but people in the field of wearable robotics who work on exoskeletons, is really what should the pattern of force be in this green parallel element in order to, for example, improve the gas mileage of a person when they walk or run. And more recently, we've been thinking more about not just energetics, but things like what should the pattern of force look like in order to improve stability or even maybe agility as well. And of course, by studying this, we can understand, again, a little bit more about the biology itself, because we can push the biological elements in the musculoskeletal system to extreme areas in their functional space, which lets us learn a lot more than we might if we were only studying steady state locomotion in the absence of devices. And the lab takes a multi-scale approach. We do some muscle physiology work on isolated animal muscles. I won't really talk much about that today, but that lets us start to explore things like sensory feedback and how devices might affect the sensory side of the musculoskeletal loops, of sensory motor loops. We do modeling and simulation with simple models like these. And then we study humans, thinking about things like stiffness or impedance around the joints, using standard techniques from locomotion physiology. And more recently, we've started to think about perturbed locomotion. What happens if you fall into a hole or if I push you from behind or the side? Okay, what I'm going to focus on today is not the stability or agility question, but on the question of economy and how devices can improve the economy of locomotion. And this is a nice slide to start with. It's a broad overview of progress that people have made in the field since about 2009 in improving the gas mileage of walking and running by adding assistive devices to the body. And what you'll see here is that moving, I guess, sort of rightward through time and downward in terms of metabolic benefit, the field is ticking ahead. We're doing well. We're getting better and better. And I don't want to get too much into the coding in here, but we're doing this in different ways. Sometimes with passive devices, most of the time with actively powered devices. We've done it now as a field at different joints, the ankle, the knee more recently, and the hip, and now we're starting to explore combinations of assistance at joints, and so far we've gotten to about a 20% improvement in the economy of walking or running, depending on which gait you're looking at. And I happen to know just from attending recent conferences that this number is now approaching about 50% reduction. Laboratories like Steve Collins's lab at Stanford, in work led by Patrick Franks and others, have shown that in a multi-joint tethered system with optimally timed torques that you can approach 50% improvement in cost of transport, which is quite amazing. What I'm going to focus on today is the class of passive devices that don't transfer net mechanical work to the person, but instead capture and transfer, you may say, recycle energy and reduce forces, but not reduce net mechanical work over a cycle. And that brings us to springs in locomotion. So a lot of our work is motivated by elastic mechanisms that we observe in biology and animals, including humans. This is a nice review paper by Tom Roberts and Mani Azizi. The Roberts lab is where I did my postdoctoral work. And so this is kind of where I began to think a lot more about the muscle level, and muscle tendon interaction in particular. And really, elastic mechanisms can improve performance in all three of these sort of areas of locomotion performance: economy, agility, which you might think of as power amplification, and then also stability, which you might think of as power attenuation. And again, today I'm going to focus on elastic energy storage and return, both in the device that I'll share with you and also kind of in the human studies that I'll share. But it's important to remember that elastic mechanisms can also be important in these other regimes. And I just want to kind of shout out some recent work by Taylor Dick, who's now at the University of Queensland, in collaboration with Laksh Punith, who's a PhD candidate in my lab, on hopping and then falling into holes. And so we've done some muscle level work using ultrasound to look at how elastic mechanisms can prevent injury to muscles when they absorb large amounts of energy in the context of cyclical movement. And eventually we want to use those data to understand or to inform how better to devise devices that can further improve the injury avoidance. This is just a fun slide that I always like to flash up as a reminder that it's not just animals, but, you know, and we're not the first ones to think about using elastic mechanisms in machines. Here are a number of examples from biology, frogs, kangaroos, and things like really large birds that use their tendons to get very economical gait are kind of inspiration for us. And then these ultra marathoners that can run, you know, 50K races many days in a row at very high speed with very little energy. And then on the machine side, robots that are using series elastic actuators or even elastic mechanisms to provide stability or agile movement without the use of over-actuated joints. Okay, so just into a bit of data now. So how do we think about this biological inspiration in the lab, and how do we bring data to bear in order to understand the system so that we can improve it? Well, the first thing we can do is what are now kind of simple and standard gait analyses. We can examine the amount of mechanical work done by muscle tendon units about the joints. And that's sort of where our bread and butter is. We think about the center of mass some, but we really kind of live at the joint and muscle tendon unit level in my lab. And the sorts of data that we get out can give us at least kind of zoomed out information about the behavior of muscle tendon unions around the joint over a cycle. If we see net mechanical energy being generated by muscle tendon units, again, using a basic inverse dynamics analysis, we might replace that function with a motor because we know that there's net mechanical demand that needs to be met. If on the other hand, you know, this would be level ground or going downhill, perhaps at the knee joint, we see net negative work over a cycle. We might look at that as an opportunity to harvest energy and either store it or charge a battery with it or recycle it at some other part in the gait cycle through physical connections or electromechanical connections. And finally, if we see kind of net zero energy behavior, we automatically think about springs and elastic elements. And the ankle is probably the focal point for that kind of joint level behavior. What we do, you guys are all familiar with this. It's standard split belt treadmill, instrumented treadmill, motion capture. Combine those, you can do inverse dynamics. But we also, we record electromyography because we, at least more recently, it's becoming very clear to us that the intensity of the EMG may do the best job of predicting the ability of a device to reduce metabolic costs. So we tend to monitor that a lot now. Ultrasound imaging lets us look at fascicle length changes in vivo, and that lets us relate performance of the muscles to the fundamental force length and force velocity curves as those relate to energy consumption. And then kind of the bread and butter measurement comes from calorimetry, which is what we're using to understand how much food energy the person uses based on oxygen consumption and carbon dioxide exchange to power movement. And really, this is kind of the mainline performance metric that we use when we're evaluating gas mileage in and out of robots. So here's what some inverse dynamics curves might look like for level ground in green, I've got joints going ankle, knee, hip here from top to bottom. And in this particular case, we studied a number of different grades. These are data that have appeared in the literature for a very long time. So we're not showing you anything new here. But we tried to interpret this data in a new way with respect to those functional bins that I described before, spring motor brake, if you will, or elastic mechanism motor harvester. And one way we think about that in our lab is through this pie chart analysis. So you can take the areas under these curves to convert instantaneous power to work over a cycle. And you can plot those as pie charts where the size of the pie represents the total amount of positive work in the top row here and then negative in the bottom. And you can further split out the positive and negative work across the joints as percentages. And what you'll note here, like any good mechanical engineer, is on level ground at 0% grade, there's about an even amount of positive and negative work over a cycle. These are fixed speed gaits, and this is walking. And what you might note is that the ankle is contributing about 44% of the positive work and about 38% of the negative work. So it's smelling like a spring. As you go uphill, the hip starts to take over more like a motor to power the movement. Negative work, of course, goes down and net positive work comes online. As you go steeply downhill, the opposite happens. You flip over the net negative, and you can see that the knee takes the majority of that negative work in steep downhill locomotion. And so if we just plot the nets coming from the sums of those pies, the data look like this. Again, there should be no surprises here based on the basic physics that have to be met. But one thing we can do is, again, like think about this analogy, right? So on level ground at the ankle, we have net zero work over a cycle. Maybe we can replace that function with a spring. Going uphill, we have a lot of net positive work, and it's especially coming out of the hip. Maybe what that means is our device should serve a motor-like function at that joint. And again, if we go steeply downhill, or actually any amount downhill, including on level ground, there's negative work to be had at the knee, potentially to be harvested. Okay, so the ankle joint is a major source of both positive and negative work during walking. And this is true not just at that 1.25 speed, but it's also true across walking speed, where the ankle and the hip kind of are the dominant sources of energy, but arguably the ankle, if you look at both walking and running together, again, is kind of the foci of energy generation and of energy cycling, really, in the human limb. And so that kind of points us like toward the ankle as a first line of attack, which is really why in my lab we focus so much on the ankle. Is we're taking the inspiration from the biological system. We're looking for the sources where we think power is needed, and we're going to try to reduce those power requirements by attaching exoskeletal devices at those joints. The other thing we can do is look further down under the skin. This is where the under the skin part of the story begins. This was work by Dom Ferris when he came to my lab in 2010-ish, when we were just starting out in North Carolina. Dominic brought the skill set to use ultrasound imaging to the lab, and ever since we've been using it as one of our main tools. And what we can do with this, you guys have probably all seen some of these sorts of data before, but I'll go through it quickly. What we can do is place an ultrasound probe over the calf. It's about the size of a pack of gum. It's pretty small. You can strap that down with Coban and ACE bandage pretty tightly. And what that gives you is a decent view of the fascicles. And what you can do then is record images and then post hoc track those images. This is a yellow line here is tracking fascicles in the soleus during walking. You can see that rhythmic behavior, and what that allows us to do is combine information about how fascicles are moving with information about how the joints are moving from motion capture to make best guess estimates of how the energy is shared at that joint between muscles and tendons over the gait cycle. Another way to think about this is if you zoomed into the ankle piece of pie that I showed you on previous slides, we can now separate that piece of pie further into contributions from elastic energy storage and return from the tendon, and contributions from force-related lengthening and shortening of the muscles. And when we do that, we also see that at the ankle joint, for the soleus and gastroc, for the triceps surae complex, that about half during normal speed walking and up to about 70% during medium pace running of the energy that's powering the movement on the positive side is coming from previously stored elastic energy in the tendon. So that signals that that's another signal that there's spring-like behavior there, right? Like there is actually a biological spring doing a large portion of the mechanical work, storing it on the negative side and then returning it to help muscle shorten on the positive work side. Okay. Now, what does that mean in an engineering sense? If you look at the length change patterns, and that's what I'm showing you here, this is length change on the y-axis over a gait cycle. The black curve is what the fascicles do. This is for walking at 1.25. And then the muscle tendon unit is in light gray here, and then the tendon itself is in dark gray. And what you'll note is that the muscles are, they're pretty isometric. You can really see this when you look at the velocities. The velocities hang out near zero for a large portion of the stance phase. And that occurs during the time when there's maximum force. So, you know, in the extreme, if you were a clutch spring system, you'd have very high forces and really zero movement of the clutch. Here I'm referring to the muscle as a clutch-like mechanism inside the muscle tendon unit. And again, that analogy is easy to make because we see strong evidence of clutch-like behavior in the soleus and gastroc during walking, where the muscle is holding a tendon that is stretched by changes in external energy of the body through stretch and recoil the tendons. High forces, low velocities are clutch-like. Okay, so we take all of this inspiration and we decide we want to build a device, and we're going to go for the passive device, because if we're going to reproduce spring-like behavior, no better way to do that than just attach springs. The other benefit is that we don't have to power any of this. If we design a clutch mechanism that is built so that it just works on the configuration of the joint, that is a configuration, it's a configuration dependent ratchet pawl system that allows the spring to be locked against during stance phase so that motion of the body can stretch it, and then natural dynamics allow recoil and push off. And then during swing phase, this clutch is loose and allowed to freely rotate so that the person can reposition their foot for the next heel strike without having to do work against the spring. Okay, so if you want to read more about this, you can go to the paper and look at the patent, and there's some videos available to really get into the mechanism. You can also get the CAD drawings for this, and a few labs now have printed it and played with it, either in undergraduate or PhD projects. So it should all be there if you want to play with this mechanism on your own. Here's what some data looks like from walking in that device. And what I'm showing you here are multiple curves because we examine the behavior of the device across a range of stiffnesses of that exoskeletal tendon, going from no exoskeleton here in black. And of course, there's no exoskeleton torque when it's not being worn. So there's a deadline 0 here for the no exo. And then as I increase stiffness from 130 to 400 Newton meters per radian, these were chosen because we measured the quasi stiffness, or the slope of the steady state moment angle curve during walking, to be around 400 Newton meters per radian during the stance phase. And so we wanted to be able to cover what we thought of as the full physiological range of quasi stiffness by adding external springs. And when we did that, things went pretty well up until a point of no return where joint stiffened so much that it caused people to start to modify their ankle kinematics. And that caused diminishing returns on our ability to generate more and more assistive torque. The person was not able to move their body into the assistance to generate torque at some point. But what's cool is for the intermediate stiffness springs, here around 180 Newton meters per radian or so, we were able to reduce the biological joint moment around the ankle by about 25%. And again, we're doing this all passively, right? So this is just basically a natural motion of the person where they're adjusting their muscle activation to accept some stiffness from the external device in order to presumably reduce the energy cost. And indeed, that's what we saw. This was a pretty exciting result, given that this was a passive device. And prior to this, all of the demonstrations of improved gas mileage came from devices that transferred net mechanical energy through motors, either tethered or autonomous. And what we were able to show here is that when we added springs of intermediate stiffness, we could get about a 7% plus or minus 3% reduction in the metabolic cost below normal walking in regular shoes. And things sort of were not as good for less stiff springs, and they got pretty bad. If you go beyond this 400 level, things really start to fall apart. People feel pain, and they modify their movements so much that compensations begin to appear at other joints, which interrupts the assistance and causes a net increase in the cost of transport. Okay, so stories so far is we took biological inspiration by making relatively standard measurements in the laboratory to understand the sources and types of mechanical energy that were being generated at different joints during movement. We noticed springs at the ankle. We reproduced a spring-like device that was able to offload moment and presumably force at the muscle level and improve cost of transport. Pretty cool. The next thing that we wanted to do is see whether or not that strategy would work across walking speeds. And so the data that I just showed you before was only for walking at 1.25 meters per second. And this was work by Rich Knuckles, who's now a postdoc in Connor Walsh's lab at Harvard. And what he did in the lab is first thing he did is he converted the physical springs that you saw in that mechanism to a tethered robot that can apply different amounts of compliance in parallel with the person using feedback control. So the idea here is that we're measuring the joint angle on the exo, and we have a software desired stiffness that we want to maintain. And the device will provide the appropriate amount of torque to enforce that stiffness relationship that we ask it to. And so if you look at some data here from the exoskeleton, you can see the exoskeleton angle against the exoskeleton torque. What we're hoping for here is sort of nearly linear or close to linear curves, because we did not program a nonlinear spring that can range across a reasonable range of stiffnesses. Here we're not going up to 400, knowing that 400 was uncomfortable for people. And we're able to do this sort of now across walking speeds on the treadmill. We can do these experiments very quickly. We don't need to build anything. All we need to do is type in the stiffness that we'd like, make sure the device is snug, and we're good to go. And what that let us do is examine the range of stiffnesses across speed. Here's what we found. We were able to reproduce the result at 1.25 meters per second. This time we didn't quite reach 7, we got about 5% reduction on average. You'll note that the stiffnesses where we found minima were different than what I reported before. And this is because in the paper earlier, we calculated the stiffness based on a linear stiffness from McMaster car catalog. We did not characterize the stiffness as the person feels it. And so there's a lot of compliance, right, between the spring and the person and the carbon fiber, et cetera, that the feedback control takes care of. So these are real stiffnesses on the person. They're real physiological stiffnesses controlled in real time. And what we note is that the true stiffness the person's feeling, at least in these data, was that minimized the cost of transport was more around 70 rather than 180. It's a much lower value than what we reported previously. Interestingly, we found no performance benefit at 1.5 meters per second for any spring. This is average data across, I believe it's 9 subjects. But at fast walking, we found the benefit return again. And this time, these were not statistically significantly different optima stiffness values, but maybe it's slightly higher for faster walking speed, you might say. I'm going to get into the mechanism for why we think this bowl-shaped curve arises at the muscle level for 1.25 data. And we are still working on that muscle level explanation for these other 2 data sets as we speak. But I suspect that there's a story there in terms of a force length and force velocity benefit, cost benefit trade-off at the muscle level that could help explain why we don't see a benefit at intermediate speeds. Happy to talk about those details later if we have time for Q&A. Okay, so up to this point, I've shown you that we can, with a passive device placed on the ankle that's effectively behaving as an exoskeletal tendon, when the stiffness is dialed in appropriately, we can get a reasonable reduction, a pretty good reduction in cost of transport. 7% is about a small rucksack, like put your laptop in a backpack, and that's about 7 or 10%. Another way to think about this is if I asked you to walk all day long without using your arms, your energy costs would increase probably somewhere around 10%. Another way to think about it is something like 0.25 to 0.3 meters per second worth of walking speed. That's about a 10% change. So it's not functionally insignificant. So we were pretty stoked about this. We're really excited. And what we set out to do kind of after those initial studies was to try to understand a little bit more about why it works. And just to start this section of the talk, I want to have people think about what an exoskeleton is in kind of block diagram framework here. So when I think about movement, I think about a motor command that hits, it's not just one muscle, but let's just imagine the calf muscle tendon complex again. Motor command comes down. There is, of course, feedback and feed forward neural pathways that contribute to that motor command. The muscle converts its kind of length and velocity, its state, and the motor command. So its full state gets converted to a force. We know a lot about how that happens from fundamental principles like the force length and force velocity curves. When there's a compliant tendon in series, the force is routed through that, and it also goes to the external environment. And this sets up a very interesting scenario for compliant muscle tendon units, which is that the muscle's ongoing state not only depends on the movement that results from those forces in the environment, but it also depends on the compliance of the tendon. So you can think of there's an internal force feedback that adjusts the length change patterns of the MTU, which come from external movement, and converts those to what the real feel of the muscle in terms of the muscle state. And this is going to become important later when we think about what a passive device does in parallel, which looks like this. There is no command coming from anywhere for a passive device. The passive device is reaction. It's reaction to the position. If you put a spring stiffness in here, the position of the body is what's determining the ongoing force of the exoskeleton, right, through a stiffness relationship. And now there's a combined force, biological plus exoskeleton, that feeds back on the environment dynamics. Okay, so when we put a passive device on someone that's merely a spring, this is what we're doing to the kind of neuromechanical block diagram. And if we want to understand what's happening at the level of the muscle energetics, we really need to think about how this block affects the state of this thing, right? And the energetics of this thing in particular, which I'm going to, in a couple slides, try to relate to force length and force velocity sorts of things. Okay. I can take a quick pause here. If there are questions now, this might be a good place. Otherwise, feel free to hold those until later, but are there questions yet?
Yeah, can I just put that diagram I'm wondering where there's another parallel elastic element, the deep fascia and the skin would both be, when talking about ankle extensors, do they,
Yeah.
they're not under exoskeleton, but they're both parallel elastic elements. I'm just seeing how you would put them into this diagram.
Yes, I think those would go sort of inside the more detailed muscle block, where I might include parallel elastic elements that are for things like fascia and extracellular matrix even. And of course, like, there's a lot of good data in the last 5 years from people who study the morphology of muscle tendon units that kind of make this line very blurry here, right? Are things in parallel or in series? Where's the aponeurosis? Where does the muscle end and the tendon begin? Does the motor command have some impact on the tendon stiffness through shape change? Things like this are not included in this very simple diagram, but certainly could be important.
What about skin, which is very much away from the muscle, but can be quite strong, particularly if it's dry?
I think the skin would come somewhere in here between the exoskeleton and body, and right where we're at the interface between the device and the body. There may be some compliance or some slip that makes the transfer, especially of energy, but you know, the forces will get there at the end of the day. It's just a matter of what displacements are involved. And those things become very important when we think about actively controlling devices, and the feedback control can really fall apart if there's a poor interface. So I really, in a passive device, it may not be as big an issue, except you may not know what real stiffness is getting to the muscle because of all the compliance between the device and the tissues. But we don't see it as a huge issue until we get to kind of high bandwidth force control of an active device, where these things like skin and other things might play a role in the interface. Were you thinking more about the physiological state of the muscle in terms of the skin?
No, I'm just purely thinking, I'm aware that the skin, whether it's moist or whether it's dry, can be a compliant or a stiff parallel elastic element that impacts, you know, certainly on the thorax, but also on the ankle, I imagine, and as in much the same way that an exoskeleton would.
Yeah, and I think that might go back to this muscle tendon gray area, where if we're thinking about, you know, my conceptual diagram here, and you'll see in the next few slides, is basically ropes. These are one-dimensional models in my mind. I'm not even thinking really about pennation angle, but when you talk about shape change and bulging, certainly there the skin can be an important constraint too that might have effects on the ongoing length and velocity of the muscles.
Go on, sorry, I interrupted.
No, you didn't, thanks. This is fun. If there are other people who have questions, feel free to speak up.
Yep. I also think that there should be much more question coming after your talk is done first. So if you don't mind to keep moving on.
I will keep going. Here we go.
Thank you.
Okay, so let's look down a little bit further into the muscle level and think about how the heck does this thing work without doing network on the person, right? And this is just dissecting down from this overview free body diagram into what the physiology is feeling. Here's the exoskeletal spring in parallel with the triceps muscle tendon unit. I already mentioned this, but the loads are getting split now between biology and exo, which means that if all else is equal, the loads on the biological system will be reduced. The fascicles themselves do work at a pennation angle, which may or may not be important because the dynamic change of the pennation angle is load dependent. And then really what we care about if we're thinking about cost of transport is the heat generated by cross bridges as they cycle, and also as they're recruited to begin with, right? The activation and then cross bridge cycling costs. And to think about those, we can really, I guess, imagine that the fascicle is representative of what might be happening at the sarcomere level. And I know that is probably not the case, but that's as far as we go on the musculoskeletal scale. We're not going to go beyond fascicles to single fibers and then down into the protein level. But we are making assumptions that the length change patterns that we see in our ultrasound images are somewhat representative of what might be happening down at those levels. And really what we do here is we use motivation from the work of Roger Crum and also back to C.R. Taylor and Tom McMahon, that another way to think about energy costs during locomotion, one way is work efficiency, right? But if you have a device and you have a movement that's producing no network over a cycle, another way to think about these things is in terms of muscle volume or force-related costs. Costs just to hold a tendon in place, for example, or the cost just to stand up without moving. These are force-related costs that are always there, even when the forces are produced dynamically. And so we want to motivate kind of understanding the mechanism from the place of active muscle volume. In order to do that, we can invoke the force length and force velocity curves as a way to kind of bring in the operating points of the biological actuator as we think about how much it might need to be activated for a certain amount of force that's required of it. And so you guys all know the force length and force velocity curve, but ideally we would love it if the muscles were always operating at their rest length here at L0, where it doesn't take a lot of activation to produce high forces, and also isometrically, or even on the lengthening side of the force velocity curve, where we know there can be advantages in terms of, again, the force per unit activation that one can get from a muscle contraction. And if we kind of rearrange this basic Hill-type force equation and we solve for the activation, we're calling this the active muscle volume that we might expect, we see that force capacity is important. So where the muscle is operating on these two curves matters, but also the force demand, how much force is needed in the first place. And I want to argue that exoskeletons influence both of these. It's quite obvious in my mind that exoskeletons can reduce the force demand if people maintain the same overall movement pattern. The forces that are required of the musculoskeletal structures are lower. And it might even be during augmented locomotion that the forces are lower, but that's what the device is there to take some force off the musculoskeletal structures. And now what's really interesting is when you have a muscle tendon unit that's got a long tendon on it, through that internal force-based feedback on the tendon, you can get shifts in the length, tension, and force velocity curve for a muscle when you put force in parallel with it. And the simplest way to think about this in my mind is if I put a spring in parallel with something that is a muscle and a spring in series, and I ask this whole thing to produce the same amount of force, some of that force is now born in the green element, which is the exo, and a reduced amount is produced now in the muscle tendon unit. That means that tendons will be on average shorter, which means that muscles, if the whole muscle tendon unit movement remains invariant, muscles should operate slightly longer lengths. And that's conveyed here on this hypothetical length tension curve. So here's where a person might be operating without an exoskeleton in this purple region, kind of on the ascending limb at reasonably high force levels. If I place an exoskeleton on them, they should travel down this curve to a lower force requirement along the constraint provided by the tendon length tension curve. And what that means is lower forces at longer lengths. And that should lead to lower activations because of an increased force capacity, i.e. a better operating point on the length tension curve potentially, if I move you to a longer length from the ascending limb, and also a reduced force demand. Okay. Was there a question or should I keep going? Nope, I'll keep going. So we wanted to check on these ideas experimentally now. So we pulled back out our tethered spring-like exo. And we did a full experiment where we studied exoskeleton mechanics and energetics over many sessions. And really what I want to focus on now is the ultrasound data. We had ultrasound running this time. And we tracked the soleus fascicles again. And here are what some of the muscle level data look like. So we just estimated muscle forces based on the inverse dynamics. And as expected, as you increase the stiffness, the forces go down. So that's a reduced demand, like I mentioned. Reduced forces at every point, pretty much in stance, and mostly in late stances where we saw effect. The other cool thing is that the hypothesis that the fascicle lengths would increase was borne out in the data. So as you went to stiffer springs, fascicles operated on average at longer lengths. And this was about a 10% shift on the kind of normalized force length curve, which I'll show you in a bit, which is significant, we think. So we saw significant increases in the lengths at peak force and the average lengths over a cycle. And with that, we did not measure the length tension curves for every individual. This is just a drawing of what we expect a sarcomere level length tension curve to look like, and how our data might map to that. And what this is showing you is that we are able to move people from the ascending limb, which is where they normally operate during walking, further toward the peak, the place where L over L0 is 1.0, by increasing the spring stiffness. And that means better economy of force production, right? You can use less activation to produce a given amount of force. And we think that helps explain at the muscle level, the local muscle level, why we see decreases in the metabolic costs for stiffnesses that are for the lower end exoskeleton stiffnesses. Now, if you look at the force velocity relationship, there's a bit of a different story. So even though we were able to shift muscles to longer lengths to a better place on their length tension curve, the velocities went the other direction. And they went the other direction in the very sensitive part of the force velocity curve, so that even small changes in the shortening velocity at the end of the stride, which you can see here, at time of peak force and later, we found increases in the shortening velocity of the soleus. And that converted to reductions in the ability of the muscle to produce force. So we think there's a force velocity penalty. That might help explain why the device doesn't work so well for the higher stiffness conditions. Okay. So if you put all this together and you examine the activation, right, the active muscle volume, or the intensity of the EMG as a proxy for that, what you find is pretty good correlation. It's not great, but remember, we're only looking at one muscle here. And we feel like if we did this analysis over many more muscles in the lower limb, that this relationship would just get stronger. But what it says is that the activation in the muscles correlate quite well with, or associated with the changes in metabolic rate that we found. And that's important because it might provide some clues if we were to do real-time feedback of a device in the future. Maybe we should be paying attention to the person's muscle activation as an input signal to that controller. Okay, so we found that changes in metabolic rate correlated quite well with changes in the muscle activation. We saw that lower stiffnesses, the performance for the lower stiffness exoskeletal conditions could be partly explained by changes in the length tension behavior of the soleus. And at higher stiffnesses, we feel like there's a local explanation at the muscle level for poor performance based on the force velocity curve and pushing the muscles to faster shortening than they'd like to be. Okay, so I want to present some really new data at the end here in the last few minutes. Owen, with this information in hand, I guess we kind of challenge ourselves to go and prove it out a little bit more. Like, is it really true that the length tension relationship and the force velocity relationship are related to metabolic energy cost of cyclical contractions? Like, how can we study that more carefully? And one way we thought to do this was using a dynamometer. And so this is work by Owen Beck in collaboration with Jordan Schrader and Lindsay Trejo, and also Jason Franz, who shares the funding from NIH for this project with our group. And what Owen did is he devised a dynamometer setup that looks something like this. It's very similar to what you guys are doing up in your group at the knee joint, except here we're at the ankle. So we have the person in the deep knee flexion, And that's in an attempt to only include the uniarticular soleus in the movement, not in the movement, but in the force production task. So we're trying to make the gastroc slack here. The foot pedal is fixed to a number of different postures, which allows us, in theory, to move up and down the length tension curve of the soleus muscle tendon unit. And then what we do is we give feedback through visual display for the subject to target a peak torque value. And again, these are what we would call isometric or fixed end contractions. So they're not actually moving the muscle tendon unit. They're just pulling on the tendon internally in these force production cycles. And so each cycle is the muscle pulls on the tendon and then releases it, pulls on the tendon and then releases it. So the load is the tendon itself. But still, we can vary the torque that's required, and we can vary things like the time course of that if we want, and the magnitude of that. And we can also, again, we can vary the kind of the fixed posture, which is what we focused on in this particular study. And these data I'll go through real quick. These are just to show that our protocol more or less worked. So we could get different moment levels by feeding back different levels of torque. We could get different levels of force as estimated by inverse dynamics. And we could do that at a range of different ankle joint angles. This is the fascicle pennation angle, which we can also monitor, but really doesn't play into the story here. And then here are the fascicle forces. And so the idea here was really to try to maintain force over these angles, which we were not perfectly successful at because of changes in the moment arms that come with posture, which in hindsight, we probably should have tried to account for. So there are some imperfections here in the protocol, but suffice it to say that we're able to vary the force and also vary the length at which the muscles are operating in each of these contractions. And that's what I'm showing you here. As we increase the ankle joint angle by pointing people into more and more plantar flexion, we're able to reduce the in vivo average fascicle lengths. Another way to say that is we're pushing people from the kind of high ascending limb down the ascending limb in different trials, okay? Moving them away from optimum length as they get shorter. Of course, this also has effects on the velocity because tendons are nonlinear and the load is changing at the same time as we're changing the posture, right? So we might be sort of in the toe region of the tendon for one trial and in the kind of linear region of the tendon on another. And so there are kind of side effects on the velocity behavior as well that we really couldn't control very well. But here's kind of the take-home message, is that we found a strong effect of the ankle joint posture, aka the fascicle average length, on metabolic rate. And what we found is that the metabolic rate sharply increased at both the low and the high force levels as we increased the ankle joint angle, aka shorten the fascicles down the length tension curve. And what's more is that the increase in metabolic costs that we found was way more than we would have calculated from those active muscle volume theoretical equations that I presented before. So there's a disconnect in our data that we cannot explain. Maybe it's force rate dependent effects. It may just be an explicit length dependent effect on how cross bridges use ATP during cycling. We're not sure, but the active muscle volume model sort of really fell apart here. It was not able to explain the market increase that we observed. So what's interesting is there is an increase. And this partly explains or helps corroborate the idea that an exoskeleton can have big effects on the energy needed for contractions by moving the muscle lengths on average to either longer or shorter lengths. But the theory behind why that works is incomplete. Okay, I have a few fun slides to finish here, which are forward-looking, like what are we up to now and next? One of them is trying to think about this idea of muscle in the loop control. So this idea of in the loop is becoming pretty popular across laboratories for good reasons. It allows the experimenter to understand causal links rather than associations between things like contractile properties and metabolic costs, for example. But also from the applied sense, if we want to build devices that are truly symbiotic, we believe in our lab that we should be looking for ways to link the behavior of our devices with physiological states more explicitly. And one way to do that might be through EMG recordings, for example, but another way might be to be more explicit about using muscle dynamics to drive behavior of robots. And this could be done to avoid lengths that are too long and keep muscles safe. It could be done to get muscles in the sweet spot of the force velocity curve during jumping, for example. Maybe try to get them around 1/3 V over V max. And for economy, it could be like I've already described, like trying to get muscles closer to L naught or into the sort of shallow passive, where the passive length tension curve begins to come online, and we know that there are energetic benefits there. So one thing that we're doing to try to make progress there is to use machine learning to take our ultrasound images to a real-time feedback loop application. And so what that means is we need to train neural networks or other types of machine learning algorithms to take fascicle data so far from images is what we're doing. We're not using raw sort of RF data, but that's another possibility. Just B-mode data at this point to see how far we can get in terms of our ability to track muscle fascicles in real time for biofeedback applications, and then maybe also for kind of slow or pseudo real-time control devices. Another thing we're thinking about is, this is a bit bigger picture, but like if you went down even further to sort of biomarkers of muscle or cartilage or bone health, and you were able to track those longitudinally over very long time scales, again, with the aid of machine learning algorithms, perhaps, could you begin to think of exoskeletons not as really augmentation devices, but instead as devices that maintain musculoskeletal health or homeostasis over longer periods of time? So the idea of these devices to improve longevity rather than say acute behaviors, in terms of locomotion. And this is the last slide. Finally, I kind of alluded to this in the beginning, but I'm really interested in the question of how exoskeletons, how they influence the sensory side of sensory motor loops. Everything I've talked about today is mechanics, right? And mechanics from joints down to muscles, great. We think we're starting to get a handle on some of that. But really, there's not a lot, if any, information out there on how devices like this influence sensory feedback. And that's important. If you're a rehab roboticist and you believe that relearning a task depends on error-based learning, it's important to understand what the error signals look like and where those error signals are coming from. And presumably, if an exoskeleton is modifying the length change behaviors of muscles, and spindle organs are inside the intrafusal fibers of those muscles, then the sensory signals are also different from what they were before the exoskeleton was donned. And so in order to get at this, we can't really do these experiments in humans yet. Microneurography is the approach, but that's very hard to do during dynamic movement, and certainly it's even harder during robotically assisted movements. So our approach to this is to try to build locomotion up on the benchtop, like in a surgical suite, where, for example, we can take a rat preparation and place this rat in a fixed stereotaxic frame, which allows us access to the spinal cord, where we can take recordings from 1A or type 2 or 1B afferent pathways, while we manipulate the muscle through sort of a haptic interface that maybe embeds the physics of a rhythmic motion, like a bouncing gait, in conjunction with a modeled exoskeletal device. So the idea here is to model the physics, model the mechanics, embedded in an interface that attaches to a living muscle, and then take advantage of that kind of virtual reality, if you will, to make these high fidelity recordings and start to get a handle on what's happening on the sensory side. Another approach to this is to use living, moving animal models like guinea fowl. And so I've started a collaboration. This is over slow bits and spurts over the last few years with Jonas Rubinson, and Zan Cox was a major player when she was in Jonas's lab, to build this look like, you know, a Harvard Vis Institute robot for a guinea fowl. And the idea here is that if we can build something that is comfortable enough, we can really examine these animals over months, potentially, in flocks that wear these versus don't, to try to understand the long-term effects of these sorts of devices on the musculoskeletal system. Okay, that's where it ends. So I'll pause there. Hopefully we'll have some time for questions now.
Thank you very much, Greg, for giving that great talk. So if you could now unshare your screen so we could see each other during the discussion period, it'd be wonderful. And I cannot see you now. Yes, now I can see you. Thank you. And I also would like to kindly encourage everyone to turn on your camera if you feel comfortable with it. I think then we could have more active discussion period. Thanks for doing it. So if you have any questions for our speaker, please use the icon raise hand button, which can be found if you go click reaction icon at the bottom of your Zoom screen. If you cannot find the button, that's also okay. So just feel free to send me a message via Zoom chat, then I'll call on you or read your question when it's your turn. Now I'll open the floor for the questions. Yes, Kun, please go ahead.
Thanks. You were talking about active muscle volume, and I feel you used that somewhat interchangeably with muscle activation. Are these really interchangeable in your mind, or are they separate things? And can you even discern them from your experiment?
Yeah, that's a great question. I mean, I think originally we made the assumption that they're similar, if not the same. And the data that we have coming in from these dynamometer experiments especially indicate that that's not correct. So there are processes that that create a disconnect between sort of the Hill model-based formulation of active muscle volume and the measured activation of the muscle. So some possibilities here are that the model that I presented might only be good for the time when a muscle is active and cross bridges are cycling. It might not account for the sort of turn on, turn off costs appropriately. That might show up in EMG as well, right? Another issue could be fiber type and also recruitment dynamics that are kind of embedded in the EMG that do not relate to the mechanics as I presented them in a Hill type model. So those are just two possibilities that we're thinking about. One thing we're thinking about doing is, in order to get at the fiber type aspects, or even the rank, you know, which motor units are firing when, whether they're slow or fast twitch, is to think about kind of wavelet or some more complex analyses on the EMG that might help us discern whether or not in our preparation we are really, you know, which fiber types are involved. And the relationship between active muscle volume and EMG may be fiber type or motor unit type dependent, I guess is what I'm getting at. And so we're not accounting for that by looking at the EMG in a lump sense.
I see.
Anyway, those are just a few thoughts. I mean, we're currently thinking about this a lot. So if you, and I know you guys are too, I mean, Tim's work with art, we're following that real close. You guys are focused really nicely on the sort of rate dependent aspects. And we've kind of, I don't know, I won't say we punted on it, but we incorporated those sort of in our active muscle volume, maybe inappropriately as a first pass. And that needs to be addressed moving forward for sure.
I see Art, you have a question?
Sure. My question is, what is the involvement of the force length curve in your active muscle volume measure? And the reason I ask is that it seems to me that, let's just take muscles at a short length, when you don't have, so that basically means that there are many cross bridges that might not overlap with actin binding sites. And it seems like that doesn't cost you anything in the sense that those cross bridges can't cycle, so then they can't consume ATP. And so of course you produce less force, but it seems like the amount of energy it would cost in an isometric case would just be this proportional to how many cross bridges you do have cycling. And so then in a sense, force length curve doesn't cost you anything. It just limits how much force, but for how much force you produce, it seems to me that it should just cost an appropriate amount of energy. So I'm just wondering, why does force length curve matter?
Yeah, I guess our thinking on this is that the probability or propensity to bind might be different at different lengths, and so even though each cross bridge, when it's bound, might not cost the same amount, you need to cycle more in order to get those number of cross bridges bound. I don't know if that makes sense.
But doesn't cycling only happen if bound?
Well, I mean, cycling happens when bound, but also cross bridges that aren't bound, right, are there and potentially in the way. And so the propensity to bind is different at different lengths potentially, right, because of the overlap geometry.
I see.
Within the sarcomere.
Okay, thanks.
Eric, please go ahead.
Thanks. Thanks for the talk, Greg. I think it's fantastic how much you guys have evolved over the years in trying to explain all these different mechanisms. My question is regarding why target the soleus so much when there's like the gastroc is also working in the very same region, and there's even like a larger, the gastroc is larger, and or the gastrocs can combine or something larger than the soleus. So do you know how much your exoskeleton and all these experiments are affecting the gastroc and how much it's affecting the gastroc versus the soleus?
Yeah, just to answer the first part, the focus on soleus, I guess, is it's easier to think about because it's uniarticular. So the simple models that we use in combination with our ultrasound data are simpler. We don't have to account for movement at the knee, for example, to estimate muscle tendon unit lengths. In terms of physiological cross-sectional area, the soleus is larger than the gastroc, actually. It's the biggest of the triceps surae, so that's another reason to focus on it. And then in terms of how the exo impacts soleus versus gastroc, the best data we have on that are changes in the EMG that we observe, and on a percentage basis, we see similar reductions in the gastroc versus the soleus EMG when we apply a given spring. Now, one thing that we have thought about and other groups have thought about as well are sort of biarticular devices that might be able to bridge both the knee and the ankle. And in that case, might focus more directly on gastroc because of the biarticular nature of that muscle. Another thing I'll note is that people tend to compensate by in the very stiff exoskeleton conditions. There's a disconnect between relative decrease in the gastroc versus the soleus EMG. So it seems like people are protecting themselves from hyperextension potentially when you give them very stiff springs by not reducing the gastrocnemius activation as much as they would otherwise. So which is interesting to us because it seems like there can be kind of a splitting in the motor control of those two muscles where the gastroc becomes protective and the soleus keeps doing what it's doing.
Interesting. Thanks.
Yes, Tim, please go ahead.
Yeah, thanks for the great talk, Greg. Yeah, my question is quite similar to Art's. So basically, you and I both observed the same effect, and you related to active muscle volume, I related to force rate. And so in our view, this cost is mostly associated, or we think that it is associated with calcium pumping. My question is, do you agree with that? Or do you think that it's also the cross-bridge cycling cost that is causing this effect?
Yeah, I mean, I think it's probably both. So the way that I didn't present this figure, but we calculated a force rate in our contractions too. And we found no difference in the force rate. And the way we calculated that was we just took the peak estimated force over the time to peak on average for a given posture. Okay, so like at short lengths versus the longer lengths. We found no significant difference in the rate, which led us to conclude that the difference in metabolic rate that we're seeing is actually some sort of length dependent thing. So I don't want to take sides, I guess, but I think that our interpretation of our own data in light of your guys' analysis suggests that there is some length dependent thing, whether it's related to cross-bridge cycling or not. I mean, I would expect that it is in some way. What the mechanism is, I don't know that yet. But there does seem to be a compartment that is for length alone, right? Based on maybe the way we've calculated rate is not. I mean, I think that's an appropriate way to calculate rate, right? We just took our force traces and basically did what you guys do. Maybe it's a velocity dependent thing. We didn't explore that in depth, but certainly the differences in posture and the differences in the spring-like load that come from the tendon could have effects on the velocity. And I showed you that, but those were small compared to the effects on the length tension curve. So anyway, I mean, based on the best interpretation of the data we have from our experiments and also from running similar analyses with respect to the rate, we're concluding at this point that there's some length dependent thing that's there.
Yeah, I just didn't really get your explanation to Art's question, in that I also would think that if you would be at, say, half of the length, or actually at double the length, you would have half the overlap. And then so if you have half the overlap between filaments, you would have half of the cross-bridge cycling possible. So the half of the cross-bridge cannot be involved and the other half can be involved. And then I would also suspect that the difference in cost would then be proportional, because there's just less cycling and there's less force, and those would be roughly proportional, I guess. But
yeah, I see the point that the cost of a engaged cross-bridge to cycle is what it is. And if I have a force requirement, I need more cross-bridges, and there's a cost to cycle those cross-bridges. I understand that. So I don't have a good explanation for, at the sarcomere level, where the length dependence would come from.
Okay, thank you.
Brent, please go ahead.
Thanks a lot, Greg. You said that when you got to the stiffer spring conditions in the passive ankle device, that subjects complain of pain, and I just didn't know if that was pain because of if it was more comfort related because of the exoskeleton skin interface, or if it was like something internal.
Yeah, no, it's mostly just that the interface becomes uncomfortable. I'll also say that people tend to move toward knee hyperextension because of the sort of the recoil that comes from the stiffer springs, tending to want to hyperextend the knee. And so there is, I guess, I don't know, there's definitely muscle recruitment in the flexors, including the gastroc, as I mentioned before, that comes online for those stiff springs that people notice. I don't know, we have never done follow-up surveys to ask whether there's soreness like in the hamstrings or anything like that, but it's mostly the interface, honestly. And then the awkwardness that comes in that rapid recoil with the stiffest springs.
Okay. And then just another question that I had was, have you tried to look into the source of the scatter, like between the extra gas mileage that different people get with the same exoskeleton device? Like, is the difference in the metabolic cost for different people with the exoskeleton, is it because they walk differently, or do you think it's like muscle architecture stuff?
Yeah, I mean, well, it's probably a lot of different things. I mean, one thing could just be the ability of different people to adapt their motor control to the device and over what time scale that happens, right? So we're learning a lot more about that, not just in our lab, but in other labs now. And it seems like, you know, our protocols, typically we reserve about 30 minutes for people to train or acclimate before we collect data. And that it's looking like that's not enough based on new data coming out of other labs that have, I guess, put people through deep learning paradigms with an exoskeleton, like many hours, up 30, 40, sometimes 70 hours of use, where that variability within subject certainly comes down over time. But also I imagine that early on in learning, people are just more variable in general across subjects. The other thing could be muscle tendon architecture. One thing that we're trying to do right now in a current project in aging is to see if we can find relationships between a user's baseline tendon stiffness and their performance in the device. That is, if there is something to these shifts in the length tension curve, and exoskeletons modifying those as part of the mechanism for reducing cost of transport, then presumably, if the device is not well matched to a particular individual and their physiology, that there might be more scatter across subjects, right? If it's not dialed in a non-dimensional sense to be optimal for that person. So those are just a couple thoughts.
Yeah, thanks.
Yes, I see Hansel, you have a question for the speaker?
Yeah. I thought maybe passive exoskeleton has benefits over active ones, that it is simpler and maybe that makes it easier to be used in some cases. But I would assume that maybe the challenge is that it cannot really adopt, or maybe its ability to adopt to be a bit limited. So first question, is it right that optimal design would be dependent on like types of movement, whether you're walking or running, or even like dependent on walking speed? And second question is how you can address that issue when you design EXO.
Okay, so the first one is the optimal, I guess, optimal spring first dependent on the task? Like, should it be different for running versus walking at slow versus medium versus fast speed? I showed some data indicating that the stiffness doesn't really change, the optimal stiffness based on a curve fit of our minimal data set shows that the stiffness doesn't seem to vary across speed, right? At low and high speeds, the stiffness for best cost of transport was about the same. For running, there's some recent work, again, from Steve Collins's lab. This is Kirby Witt's work that explored the spring solution at the ankle using an emulator similar to what we did here in running, and found that they could not find a benefit of supplemental ankle stiffness in running, albeit pretty low running speeds, only 2.7 or so miles per hour. But So those are some data points that I can point to that suggest that, maybe at least for walking, it doesn't really, once you get the spring right for one speed, it's good for others. Why it doesn't work at that intermediate speed or so far for running at slower speeds is unclear. And then the last thing I'll say is, if you just optimize the pattern of torque to get the largest metabolic benefit, the solutions don't look spring-like. Like, you can do a lot better by transferring mechanical energy from a device to a person. Whether or not you can do that in a portable framework, that remains to be seen. It's hard. Most of these data are coming off tethered systems where there are large motors on the shelf that are producing what we know are the optimal torques. But those torques may not be achievable in a portable system that's worn outside the laboratory. So those are some responses to the first question. And now I forget what your second question was. Can you say that one again?
Second question was what to do with passive system if you need to, say, adjust stiffness or something, some design parameter depending on your task. So.
Oh, like, is there a relationship that's task dependent? Well, maybe if there is a muscle level mechanism, like we've shown some evidence for here, maybe there is a way to sort of back calculate based on some understanding what the muscle dynamics look for different tasks, what the stiffness should be. But yeah, we haven't worked backwards in that way yet. We haven't even tried to. But that might be one approach to kind of either from a simple model work backwards to what the projected best stiffness might be based on the muscle dynamics, or another way might be, again, to just close the loop around fascicle dynamics and try to push fascicles to behave either on the length tension or force velocity curve in places where we know contractions might be more economical.
I see, Walter, you have a question?
Yeah, thank you very much, Greg. I enjoyed that very much. You made me think a lot. That's always great. I was wondering, in the intermediate speed in walking, where you didn't find a reduction in the metabolic cost, 1.5 meters per second, that's, roughly the preferred speed of human walking. It's also roughly the optimal cost of transport speed for walking. And I was wondering, could it be that we are so optimized at the optimal speed that we really like and that we have already minimized our metabolic cost, that you can only improve the metabolic cost for situations when we don't walk in the optimal way?
Yeah, I think that insight is how we're thinking about it as well. So if you think about where muscles use the length tension and force velocity curve at 1.25, they're sort of down the ascending limb, not at the optimum point. And the force velocity is close to isometric, but not, of course, not perfect. And it could just be that the way in which the exoskeleton pushes you works for muscle dynamics to push you to longer lengths for slower speeds, even though there's a force velocity penalty. And at the fast speeds, perhaps there's a force velocity benefit and the length tension is not working as well for you, because at fast speeds, you'd expect the muscles to be shorter, higher forces, shorter muscles. But we think maybe we're actually supporting better velocity profiles at those fast speeds. And in the middle, maybe you can't address either. The exoskeleton does not give you either a force length or a force velocity benefit. At least that's sort of the local muscle level explanation that we're thinking about and looking to test. Of course, there may be more global changes across joints at the limbs too that help explain this. But I think you're right that perhaps we're so well tuned already at that intermediate walking speed that there's not much room to improve.
The other thing that I was thinking is, muscle force goes up during walking, in the soleus that you were talking about, and goes down. And we know that stiffness of the muscle is essentially directly proportional to force. and therefore you have a continuously varying stiffness of the muscle. And I was wondering, wouldn't it make more sense that if the exoskeleton stiffness would somehow match the muscle stiffness profile, because that's continuously going from incredibly soft when the muscle is not activated to incredibly stiff when the muscle is, has a high force.
Yeah, and I think that's one thing we've thought about too, is something like impedance blending, where we are paying attention not just to fascicle lengths or force, but the coupling between those, or the impedance. And perhaps this is again thinking forward, if you were to do something like that, EMG might be a reasonable indicator of a better indicator of stiffness than either length or force alone. And another way to think about this might be other imaging modalities like elastography that could give you sort of a real-time bead on what the stiffness of the tissue, of the muscle tissue, is like, and use that as an input to a controller that would then modify in a variable stiffness manner the exoskeleton's action over the cycle.
The other thing that I wanted to quickly chat about is, the force-length relationship and the increased cost that you seem to be seeing at shorter lengths and for your fixed end attendance there. And I could see a variety of reasons why that potentially might be, maybe not the full extent, but having measured fixed end contractions in cat medial gastrocnemius, I know that at very long lengths, the fascicles essentially don't shorten at all, whereas at short lengths, they shorten by about 10 millimeters, which is about, more than 30, almost 40% of their length. And I would assume that an enormous amount of shortening that you have at the short lengths, where you have little, resistance from parallel elastic element compared to a longer length, that shortening probably would affect the force, the metabolic cost that you have at the different lengths for a fixed end contraction. The other thing, is also that we know that sarcomeres like to be at a certain resting length. And, in frogs, for example, 2.2, in rabbits is about 2.4 microns. And if you make the sarcomere shorter, they go back to that length when you relax. So we think, and other people think, that there's some resistive force when you go too far down on the descending limb. And of course, if you had to overcome some resistive force by compressing the myosin filament and other structures, then of course that might potentially cost you additional force, because the external force might be the same, but the internal force that you have to produce for the same external force could easily be greater. And, and a lot of people have speculated it would be greater at shorter lengths compared to a longer length. So I think there's a couple of things to potentially, talk to your observation that you find a greater metabolic cost at the shorter lengths.
Yeah, thanks for the idea that force is not force, because there may be other mechanisms at play, like compression, that we don't often think about, and the simple models we have in our mind those are good to think about. Thanks for that.
The other thing that I was thinking, I'm not sure, but how often people think about the force-length relationship and the force-velocity relationship at maximal levels as just kind of a scaling down. At 50%, you multiply everything by 0.5. But we know that that's clearly not correct. You get this tremendous shift and change in shape of the submaximal force length relationship. And I didn't really think through if that might possibly explain part of your thing as well. But I was wondering if you had thought about that, and if you had implemented that, a 10%, 20% activation force length relationship is very different in shape than the maximal one.
Yeah, I'm glad you brought that up, because we are, and there's some nice work by Anthony Hessel, and I think I see Brent Ratteri on the call here too. So others are starting to think now about, and I know you have too, Walters, in animal models especially.
Well, Rack and Westbury did that in 1969, so so it's an old idea, So.
But it's hard data to get in humans, and we're starting to see more data coming out of human preps that suggest this rightward shift. And some of our own data that we're acquiring now in young versus old adults to get activation dependent length tension curves show that rightward shift. With respect to the exo, what it means is that as you shift rightward, you're chasing you're chasing a rightward shifting L over L naught, right? Instead of it, and so if those are moving at the same rate, depending on how the activation dependent shift and the exoskeleton dependent shift trade off with each other, you know, it could be that the physiology leapfrogs the exo or vice versa as you go down. And so we don't know yet. We don't know, but certainly they're both moving. So it's a moving target. And how that affects the metabolic cost on the shorter end, I haven't thought about that one yet. But it's certainly happening, right? There's a shift.
Just the last thought that I had, you know, because you measure metabolic cost, you know, with oxygen uptake, and obviously that's a generic measure of the cost that you have. And you know, for example, I'm thinking about sport. I don't know anything about exoskeleton, but in certain sport, people actually use a technique that costs more metabolic cost, but it's locally less fatiguing. And I was wondering if I've ever looked at how exoskeleton might potentially, you know, reduce metabolic costs, but might at another joint induce local fatigue that actually would make you get more tired faster, even though the metabolic cost is little. I'm thinking about cross-country skiing and double poling, which is very efficient from a metabolic point of view, but it makes your shoulders tired very quickly because it's a very small muscle mass, and therefore people will actually not use that technique all the time, even though metabolically speaking, it would be the best.
Yeah, that's another good point that we haven't explored yet. I guess one thing that we have started to think about fatigue as another way to evaluate performance. The measurement of fatigue is tricky, right? I mean, I think you can ask people how tired they are. You can look at the sort of the frequency components of the EMG, perhaps. Do you have ideas on how you would measure that?
Well, in sport, people have done it, let's say, with the shoulder muscles in double poling, and looking at that, they just took local blood samples and looked at the lactic acid in the specific muscle. So with a little puncture, you might get at that. I'm not sure.
Yeah, or even just examining sort of the ability to reach an isometric force level pre and post exercise with or without the exo could be one way to get at the force related aspects of fatigue, I suppose. But yeah, we definitely haven't done any pinprick physiology yet, that's for sure.
Yeah, and people are fairly good at telling you, you know, if they get tired at one joint more than another. Like, for example, I remember I was once in a study where I had to run as a heel foot striker and as a forefoot striker. And when I had to run as a heel foot striker, which is not my preferred technique, I got really tired in my knee joints. And, you know, and I think people are fairly decent at telling you if maybe fatigue shifts from one joint to another. But anyway, that's another thing. Great talk. Enjoyed your talk very much. Thanks, Greg.
Thank you.
If there is no last question, I would like to thank Greg again for being willing to give a talk and share his work with us. For the next week's seminar, we will hear from Danielle Whittier. The title of our talk is When Will You Fracture? An investigation into predictive fracture modeling using machine learning and advanced medical imaging. I hope you have a wonderful evening and have a wonderful weekend as well. And I'll see you all next week.